I'm Dr. Khushnudur Rahman, and today we are going to discuss the mitosis. Mitosis definition, feature, significance, cell division. So let's start it. The term mitosis was coined by Flemming in 1882. Mitosis refers to the spindle using nuclear division which produces two identical daughter nuclei from the parent nucleus. Since mitosis occurs in somatic cells, it is also known as somatic cell division. Features of mitosis. Mitosis leads to production of two daughter cells from a mother cell in each cycle of cell division. In other words, nucleus divides once in each cell cycle. The daughter cells are similar to the mother cell in shape, size, and chromosome complement. Since the chromosome number is the same in daughter cells as compared to that of mother cells, this is also known as homotypic or equational division. In plants, mitosis takes place in somatic organs like root tip, stem tip, leaf base. It leads to growth of vegetative parts. The complete process of mitosis consists only one homotypic or equational division. Now, segregation and recombination do not take place during mitosis. Remember that. The segregation and recombination occurs in meiosis, cell cycle. Now, as compared to the cell cycle, the period in which one cycle of cell division is completed is called cell cycle. A cell cycle consists of two phases, that is interphase and mitotic phase. Interphase is generally known as DNA synthesis phase, and the mitotic phase refers to the period of nuclear division. The time required for the completion of cell cycle differs. Interphase is generally known as DNA synthesis phase, and the mitotic refers to the period of nuclear division. Duration of two phases of cell, one interphase. Interphase consists of G1, S and G2 phases. G1 is the resting phase, S is the period of duplication, and G2 again is a resting stage. G1 is a pre-DNA replication phase. Dear student, that this phase between telophase and S phase, this is the longest phase which takes 12 hours in Vicia feba. Protein and DNA synthesis takes place during this phase. Protein in a mitotic cell cycle periods in a mitotic cell cycle. This phase comes after G1. The chromosome and DNA replication takes place during this phase. This phase takes less time than G1. In Vicia feba, it takes 6 hours. G2, this is the post-DNA replication phase. This is the last stage of interphase. Protein and RNA synthesis occur during this stage. This phase also takes 12 hours in Vicia feba. Broad bean plant is known as Vicia feba. However, time required for completion, all the stages differ from species to species. Mitotic phases. PMA training. The M phase leads separation of replicated DNA into daughter cells, nuclei without recombination. The daughter nuclei have the same chromosome combination in as to the parent. The M phase consists of 4 stages, that is Pakistan Military Academy training, PMA, prophase, metaphase, anaphase and telophase. Different stages of mitotic cell divisions are these. Remember that you can remember these stages by this way. Prophase starts immediately after G2 of interphase. Chromosomes look like thin thread and uncoiled prophase but become short and coiled in more distinct during mid-prophase. In the late prophase, chromosomes appears more conspicuous, short and thick and longitudinally double. The two chromatids of each chromosome held at centroma are visible under light microscope. The molecules become smaller in size. The nuclear membrane and nucleus disappear at the end of prophase. Metaphase. The phase begins after prophase. The spindle tubes are formed and chromosomes are oriented in the center, or equatorial plate. Chromosomes are attached to spindle fibers at the centromers. Chromosomes are clearly visible at metaphase. Sister chromatids of each chromosome are joined together at the point of centromer, but their arms are free. So the next one is anaphase. Anaphase, this is the phase when chromatids separate at the centromer and move towards the opposite sides of the poles. Chromatids of each chromosome become free at the centromer, but each chromatid is attached to the spindle tube. These chromatids suddenly move apart and goes to one pole and the other towards the other pole. After separation, each chromatid become a chromosome. The next is telophase. When chromosomes reach the pole, the last stage telophase begins. The spindle tubes disintegrate. A new nuclear membrane is formed at each pole covering the chromosome. The nuclei also reappears at each pole. And longer by uncoiling and unfolding and look like a single thread under light microscope. Then the nucleus enters interphase. Among all four phases of mitosis, prophase takes longest duration. That prophase is the longest one. Remember students, after that duration in different stages of mitosis, we will discuss cytokinesis. The division of nucleus is known as karyokinesis. It is followed by division of cytoplasm, which is known as cytokinesis. The division of cytoplasm into two daughter cells may take place in two ways. In plants, the division cytoplasm takes place due to formation of cell plate. The formation of such plates begin in the center of the cell, which move towards periphery in both sides during the cytoplasm into two daughter cells. In animals, the separation of cytoplasm starts by furrowing of plasma lemma in the equatorial region. This results in division of cytoplasm into the daughter cells. Genetic control of mitosis. The mitotic cell division is under genetic control in all eukaryotes under normal conditions. Important evidence in this connections are in eukaryotes under similar environment conditions, cells do not divide continuously. They sometimes divides and sometimes do not divide. This indicates that an end of mechanism must be genetically controlled. Some cells divide more frequently than others. For example, somatic cells of an embryo can undergo a larger number of mitotic division than a somatic cell of an adult. The difference in mitotic division is also due to genetic control at two different stages. The cells divide abnormally may result in abnormal tissue growth. So the position of mitotic spindle is genetically controlled. The spindles are formed in controlled manner. For example, in water snail, the direction of coiling, left-handed or right-handed, is determined by the presence of an allele. The replication of each chromosome precisely into two chromatids during three stages of interphase again provides strong evidence that mitosis is genetically controlled. Significance of mitosis. Mitosis play an important role in life of living organism in various phases. Like after fusion of male and female gametes, zygote is formed. Mitosis is responsible for development of zygote into an adult organism. Mitosis is derived from two Greek words, mitome and osis, thread division. It results in the similar daughter cells like the parents. Mitosis is essential for normal growth and development of living organism. It gives a definite shape to a specific organism. In plants, mitosis leads to formation of new parts, that is root, leaves, stems and branches. It also helps in repairing of damaged parts. In case of vegetative propagative crops like sugar cane, sweet potato, mitosis helps in sexual propagation, asexual propagation. Mitosis also leads to production of identical progeny in such crops. Again, next point is mitosis is useful in maintaining the purity of types because it leads to production of identical daughter cells and does segregation and recombination to occur. In animals, it helps in continuous replacement of old tissue with new one, such as gut epithelium and blood cells. Please like, share and subscribe. Thank you. If you have any question, you can ask.